the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Hello and welcome to a special podcast recording highlights of the UK Dementia Research Institute annual Connectome Conference. Hello, I'm Dr Anna Mallach and I'm from the UK Dementia Research Institute Centre at Imperial. And I look into the role of support cells and how they can contribute to Parkinson's and the progression of this disease. So for those of you who don't know, Connectome is the UKDRI's annual conference, which means that at the end of every year, or towards the end of every year, over 600 UKDRI members from across the institutes are coming together in different every year in a different city across the UK to talk science and meet up. It provides a very important opportunity to connect with colleagues from other centers, other locations. And today we're giving you some insights into what was presented over those 3 days that happened last week. Before we jump into the highlights, let's meet our guests. Tom?
Hi, yeah. So my name's Tom. I'm a researcher from Imperial College with the CR&T Centre, the Care Research and Technology Centre. And I'm with Paul Fremont's lab, and I have a background in engineering, and the rest of the team are all synthetic biologists. And we are developing a device which detects for urinary tract infections for people living with dementia. It's a home testing device that we're trying to provide to those so that we can mitigate the need for symptom recognition and enable home testing for quick detection.
Interesting. Beth?
So hi, I am Beth. I am a postdoc at the University of Edinburgh, and I've really just recently joined. My supervisor, Suzanne Van Belu, moved from Boston to Edinburgh, and we're part of the new British Heart Foundation Cardiovascular Dementia Research Centre. I think that's right. I'll check. I'm working on a disease called cerebral amyloid angiopathy, which is where amyloid protein gets deposited around blood vessels within the brain. So I do lots of cool research into that.
Finally, Dayne.
Hi, I'm Dayne Beccano Kelly. I'm a group leader here at the Dementia Research Institute. Specifically, the geographical location of this one is at Cardiff University. And I work on synaptic dysfunction in Parkinson's as a way of identifying early therapeutics and how this may shift and change over time to identify basically the most efficacious therapeutic strategies we can do at each phase of the disorder.
Amazing. So now that we've met the guests, for those who haven't heard one of our conference podcasts before, this is how it works. So we will each take turns discussing an interesting talk or poster that they attended at the conference that really made an impression in whatever way. We'll loop around a couple of times so you really get a sense of the broad things that happened at the conference. So Beth, let's begin with you. What stood out for you at the conference?
So there's a couple of things. So because it was my first Connectome, I was just really surprised by the size of it. I think I sort of didn't realize the scale of the UK Dementia Research Institute and, the vast amount of science that everyone sort of studies. I'm a blood vessel person, so it's really easy for me to just stay in my blood vessel field. So it was really nice to sort of learn more about the genetics and things like that people study. So that was just my first little thing. But for me, the main things that really stood out at this conference, and I feel a bit bad for saying it first, is the all the lived experience sessions. I think it was really fantastic how the start of the conference of the first day, we had the lived experience conversation with our guest Rory Capon-Jones, who's one of the hosts of the Movers and Shakers podcast, and he talked about his own experiences with Parkinson's disease, and he actually asked questions to some scientists on stage, And from that, he talks about, the importance of communicating with people living with Parkinson's disease and communicating during those clinical trials, and especially the importance of using sort of the correct language, like making our complex science make sense to people. I think that's, something really important that all researchers have to sort of focus on when talking about our work, and a sort of the focus on when there would be treatments, keeping hope but not overpromising. I thought they were just some really lovely take-home messages from that sort of conversation. And then, still going on about the lived experience sessions, I really also enjoyed how the conference sort of ended with another fantastic lived experience session. It was totally different to the first one. In this one, Charles Sabine, who represents the Hidden No More Foundation, presented a really moving talk. He narrated sort of his own experiences with Huntington's, and this was sort of you saw videos from his career, and he talked about his family, and the hope that was now here with the new gene therapy trial that we've also talked about, which I'm sure somebody will talk about in this podcast. But they were just particular for me.
Yeah, and I think particularly with this mirroring of having Sarah Trabisi give the science talk just before about the therapies, and then I think that was timed incredibly, and then having Charles talk right immediately after, not as a scientist, and coming at this from a very personal and such different angle. I think that was incredible, because I think as scientists we want to develop these cures, and I think we're often so siloed in our little science boxes that it was a beautiful reminder as to what we're working for.
I also thought that Rory Cashan-Jones's section was fantastic. He had come here to Cardiff previously, and we met with him individually, and he just was so sharp and was great at translating the science into very good and effective audio bites, rather than it just sounding cool. They were actually accurate, which sometimes may not happen when we're translating data to the media, the main screen. So I also really like that, Beth. I thought it was really good.
Yeah, I really liked his talk as well. I think it's just always so compelling seeing the people that are actually affected, and hearing, getting to engage in them and have meaningful discussions. And like you said, just not being lost in your day-to-day research and seeing actually where we're going with our work.
For sure. And I think kind of reflecting on maybe previous Connectome conferences, I thought this one was amazing that it had this focus on the lived experiences. So in a way that was kind of one of the feedbacks that I gave, this, maybe we should keep this up. All right. Tom, what was one of your highlights?
Yes, I mean, Connectome's always a bit of a funny one for me, because I'm from an engineering background, and obviously it's very focused on neuroscience, and it can be quite challenging. But in the same breath, there's so much to learn on that basis, and it just highlights how multidisciplinary the whole thing is. But I really liked. It was one of the first talks. It was Malcolm McLeod, and he's a professor of neurology and translational neuroscience at Edinburgh. And I mean, for anyone who saw it, he's super entertaining, very funny. And his focus was much more on how researchers actually conduct themselves and how to really push for sort of good quality research. And while that sounds quite obvious, it's just not really discussed and that much for early career researchers, I've found. And a lot of the emphasis is on pushing for papers and getting into good journals and things like that. And he actually spoke in one of our centre meetings a few months ago. Yeah, just again the same thing. Really thought provoking. Really makes you think about the quality of your own research. Very, very introspective. Yeah, just talking about how personal biases of researchers can really steer your research, and how to sort of avoid those from creeping in. So yeah, I always quite like his talks. They make me feel very reflective.
That was during the Early Career and Technologies Day, wasn't it? So that's the day, for people who don't know, that's the day we have before Connectome. So this case on Monday for the early career researchers and technologists, which again, I think is a wonderful opportunity to kind of bring everyone together in a room, maybe without the scary group leaders.
Yeah, I agree. with Tom. I think Malcolm's talk was fantastic, like super engaging. I think I heard so many people talking about it afterwards. And like you say, it's just something that, is definitely needs to be talked about, but it's quite hard to talk about, and it's hard to talk about in a really engaging manner that people don't feel, he just explained it in such a great way. And it wasn't just, like you say, about going in these top tier papers. It was about being, having integrity and doing the best research you can. And I think to hear it from someone so high up and so esteemed is, really helpful for, any early career researcher.
You both just mentioned it there as well, which is that there were quite a few neurologists or practicing neurologists that are also doing research. And that from my team and others that I've spoken to about it seems to have come across that that one of the best things about this particular connectome was that there seemed to be a really good breadth from basic science, as we term it, all the way through to, again, you just said the patient engagement as well, or the people with dementia engagement, and then neurologists. And it just felt like there was a really good balance this year. So you can see how it can move from one phase to the other. And that's been said a number of times, at least here at the Cardiff DRI. But you guys have just mentioned it as well. So I think that's been particularly good as well. And I'm glad it was they were so engaging, and that you guys found it engaging as well as, myself and everybody else. It was really good. It was really good.
Amazing. So now that you're already talking about what you really enjoyed, Dayne, what was one of your personal highlights?
I thought all the plenary speakers were really good. So it's not to say that they, like, anyone in particular was better than the others, because I actually, from personal perspective, really liked Erin Schumann's talk, because it worked on synapses. I'm not sure, can we classify Sarah's as being a plenary talk? It wasn't specifically gated as well, but she just rushed back from her BNA award ceremony, and so I feel like it had quite a lot of billing leading up to that, and that was also really good. But I thought Christian Haas's talk was fantastic. I thought it was really good. It was quite refreshing because he himself, I think by his own admission, was like, I'm coming towards the end of my career now, and he just kind of came out and was just like, I don't think microglia are the initiator of Alzheimer's disease. And he caught like almost a sharp intake of breath from some people in the audience, and I was a bit like, okay, this is going to be a good one, because I've seen him give talks before, and he's always really good. But I thought it was quite nice to maybe start off with what could be perceived as a controversial statement, perhaps, to someone in the crowd, but then, back it up with all of the evidence that he's had over time. And he gave a lot of, not only was the work really good and talking about the fine balance that's required between, like, microglial control within the brain and whether or not it's attacking aggregation of amyloid beta, or whether or not it's maintaining the balance, or whether it's causing detriment or keeping everything in check, was there as well. But he also gave really insightful snippets as to how to manage the career. I don't know if anybody else sort of picked up that he was talking about interactions with pharma and industry, and the best connections he's ever had, and maybe huge pitfalls that he'd interacted with, and the things that to do and perhaps not to do. And he interspaced it really well with the idea that, microglia may not be the initiator, but may well be a driver and modulator of the disease process, and affecting penetrance and affecting the manifestation. And so I just thought it was just a sort of a really wonderfully well balanced talk in giving both insight into career as well as insight into the disease state. And so I just thought it was I fanboyed a little bit and was just like, man, I hope I can be that cool when I'm getting to the end of my career, because it was really good. And I was like, yeah. So I was like, yeah, it was, I thought it was fantastic and just really, really well delivered. And the science in it was so well executed, as you would expect from Christian Haas as well.
Yeah, but in a way, I had the similar feeling, because I feel like I've worked on TREM too. I was quite familiar with the science. And so what I found really interesting was how he approached his career, and not just like science for the sake of doing science, but also thinking about it strategically, and kind of his shift towards the microglia. He was like, I had 10 years left, and then we decided to look at the microglia. And you're like, it's the art of doing science, as well as doing science.
Doing it well and having a sort of a clear understanding of, like, he did it so methodically as well. And it's sort of like, it seems like that the way he presented it, and in hindsight, but from my impression of his work throughout his career, it does seem like he built the fundamentals up and then found something and then expounded upon that and then found that, and it was just so well built. And so yeah, I'm not going to start fanboying again, but it was really good.
No,
It was really good. So I thought it was great. So that's one of my highlights. And it's not to say that sort of Erin and Sarah's work wasn't beautifully well presented as well. It just struck me personally, from my personal point of view, that it was just such a beautiful balance in the talk. So it's good. I thought that was really great.
Yeah.
I think it was also super accessible. Like, I know a little bit about TREM2, but I'm definitely not an expert in TREM2. And I think the way, his graphics on the slides, the way, he built up that information, I think it probably allowed people who didn't really know much about microglia and TREM2 to actually access some of that information, because, it can be quite hard when you're at these conferences. And if it's not your sort of research realm, we're all quite niche. I think when it's presented that way, it is just like more achievable for everyone to kind of like understand the concepts of it.
Yeah, you're absolutely right. And I think in similar vein, one of my conference highlights was actually Erin Schumann's keynote on the synapses, because I'm not a synapse person. I'm kind of starting to dabble in it. But she just started out her talk with this like beautiful rotating image of just like a neuron and all of the projections. Cool. And it was so, even as a neuroscientist, just seeing that visual, and she went very deep into the structure and whatnot. So my team have been like fangirling, boying over that for a really long time now, and I have to try and force them not to do any structural EM now, because they were like, yes, structure, and I was like, oh god, no, we there are many steps we have to take before we can get as cool as Erin, please, people.
No, yeah, it's going to take several years of dedicated work. I know that. Yeah, my PhD student wanted to go and talk to this. Erin, if you're watching this, we'd love to collaborate with you. I don't know if that was just a shameless shout out. This is my PhD student tried to find you directly after this, and we couldn't find you. So, but yeah, the work was great. I agree. It was really good.
It was really, and again, like really accessible, because by the end, like all of the like really nitty gritty of the findings and the ribosomal changes at synapses, I think that could have immediately gone over my head. And yet kind of she slowly built it up that by the time she got to that point, you're like, yeah, well, of course. That makes sense.
Yeah, I know this.
Yeah, exactly. Well, this doesn't feel like a reach anymore to kind of say, of course, they within 10 minutes, neurons or ribosomes within the synapse become so stressed that they shut down. And by the end, you were like, yes, that's, now I can go read the papers and maybe understand slightly more.
Yeah.
But what I also really, then she kind of also highlighted, the website that they had on like the synaptic RNA versus the cellular RNA. And I think that just as a kind of nod to like data sharing and making data available and accessible in a way that, everyday scientists are interested who don't necessarily have a background in bioinformatics can access it. I thought that was just one of the many things that I really enjoyed about her talk.
Really good. Really good.
Right. So we've done the first round of highlights. I think we're going to go around again. So back to you, Beth.
Yeah. So I think there was honestly just so many really cool, exciting talks, but I have to sort of highlight we had a whole session on introducing the British Heart Foundation UK Dementia Research Institute Centre for Vascular Dementia Research. That's the correct. When you move somewhere new and you have to learn all these new acronyms. And I loved that session because, it's a new sort of centre. It's not a specific place. The idea is it's the whole institute as well. We've not done it on geographics. So there's people from Oxford, there's people from Edinburgh. Where else?
UCL, right?
UCL, there we go. So it's really nice. And this was a great talk, great session. We learned about oligodendrocytes, CAA. We learned about brain energy supply and blood-brain barrier models, in vitro models. And I think it was a really nice way to sort of introduce the centre to everyone and, give an idea of the work that some of these scientists do, and then, hope for, collaborations from people. Because a couple of these scientists are new to the field. So my supervisor, Suzanne Van Baelu, she's moved to the DRI. We have Mutaz, who's at Oxford. We have Rakesh Rajani, who's at Edinburgh. And then we have Catherine Hall, who's at UCL and also at the University of Sussex. So I think it's just a really nice, and this is headed by Dave Atwell, who's at UCL. And I think it was just a really lovely introduction to, something that I'm really passionate about, which is blood vessels. I love blood vessels.
Love it. I love it. I love it. Really good.
Yeah, one of my highlights as well was specifically Catherine Hall's talk. I thought it was just very interesting. Talking about the oxygen supply and how that affects the brain and memory, and I think from my background, she managed to explain it very well. And I was able to follow through a lot of it, but yeah, just interesting how like sort of subtle reductions in oxygen, therefore energy and therefore energy to the brain just have a real impact on memory, and how this can show up very early for the disease and more obvious symptoms. But I thought that I found that quite interesting. It clicked for me that it links with some of the work that's actually done in our own centre by Dirk Jan. He focuses a lot on sleep. I don't know if you guys saw his talk as well, but he's spoken before about sleep apnea and how that obviously has big reductions in oxygen while you sleep, and obviously affects the energy supply to the brain. But just interesting thinking about those two separate sort of indicators that they both can just lead to memory problems specifically.
Definitely. And I think this is why, for example, the new Center for Vascular Research is such a good addition to the DRI, because I think we have a lot of DRI researchers who kind of dabble on the edges of kind of vascular dementia research. And so it's really nice to kind of say we have this concrete center now with our 4 dedicated group leaders, but we also obviously have a lot of other group leaders in the DRI who are doing a bit of research on that, and kind of just giving them the bigger space now to collaborate and do something. So I thought, I definitely, with our center here at Imperial, there was a lot of discussion afterwards of like collaborations, maybe even with Suzanne specifically, and doing a range of those different things. So I think it's a nice addition to the DRI, and I think we'll add a lot more than just 4 group leaders and David Atwell, I guess 5 group leaders.
Yeah, I agree. There are already so many people who do blood vessel research, endothelial cell research, astrocyte sort of research, who have done amazing work on that already. And I think you're so right. Just the addition of the centre is I think it's just nice for everyone who is, interested in blood vessels. And like you say, people who are sort of on the cusp of it. I think it's really nice to, there's so much, we need so much more research in that area. So it's really nice that there is this, the funding for this new centre.
Right. So I guess, Tom, you just talked about your highlights. So we're back to you, Dayne. Another highlight.
So I think it's going to sound a bit weird. The breaks were really good. So no, but specifically, and I say this in jest, but also I'm serious, because you just again briefly touched on it. It's really nice to sit down and have conversations with your peers, with next generation of scientists, and actually come up with some new ideas and really spitball them based on X talk you've just seen that stimulated ideas and has led you to maybe continue the conversation as you're coming out of the auditorium, and then sitting down and sort of really shooting it out and really starting to come to a conclusion that you possibly could work on something together. I had a really good chat with a few people from this, including Marco Brancaccio, and hopefully we're going to do some new innovative work together. And it's all come from just sort of sitting down and working through some of the ideas and really trying to hash it out. Could we actually do this? What would we need first? Is there preliminary data we can gather? Does this serve both our needs? Yes, no, maybe. Can we really work this through? And so, yeah, I've really valued that ability to have the period of time where you can sit down and really sort of chew the fat around the science, especially when you did have, talks like Christian's, who was being a bit controversial about something or had said a controversial statement. And so it sort of drove the conversation, the discussion that you could have, and sort of really generated that ability to have, a really raw chats about data, which can be really sort of stimulating. And so I do sincerely mean it, although it does sound funny, that the breaks were really useful in between all of those to really sit down. And I think there was the ability to do that. There was that room way at the back that had tables that people suddenly found when we were trying to eat dinners and lunch. But it was kind of there, we found ourselves in there and chatting away a few times. And so that was really, really powerful. I'd say another thing, sorry, just that was really good was I know it's general, but the in the area where we had the poster sessions was the table. And for instance, the dementia researcher section was really good for me to voluntold some of my students and postdocs to go and chat about their science and get it out there, because I think that the fact that we do have so many outlets for showcasing the work via different platforms, YouTube Shorts, audios, podcasts, etc., it's really powerful for getting our data, but also exposure of the ECR's data sets. And I think that's really important personally. And I pushed forward Gloria Chanagia and my postdoc and Shika Kataria and Shinzaid Mukhtar to put their either audio or minute shorts, and those are already up online on like LinkedIn and YouTube, and talking about their data in sort of metabolism, Parkinson's, and also looking at lysosomal dysfunction, and being able to showcase it in sort of different styles, shorts, audio, where you're answering questions, kind of gives access to what we're doing. Because I'm really a big proponent of sort of demystifying the science that we do, because I don't think it serves anything to keep it sort of, oh, you can't know about this, we're just doing science things, you don't need to look behind the curtain sort of idea. It's much better to just be out there with all the information for various reasons, but not least of which because we're doing this for, the populace, and so they should know what we're doing. So I think having all that data out there in these different mediums is really useful, and so it was really nice to have that opportunity in those spaces as well. So I really love that.
That's good. And I think talking about the ECRs, or slightly more ECRs, one of my highlights, now that we've talked about, the big keynotes, was actually the parallel sessions. So I thought it was so nice that for a couple of moments at the conference, we could break up into two smaller groups, which also meant that for at least the session that I was hosting on data and digital, which means we moved into the room upstairs. So it wasn't the big scary auditorium, it was a smaller contained room. And there listened to maybe, more the ECRs talk. And I thought that was really powerful as well, to kind of hear from the people leading the research and doing the research, what they were doing. So one highlight for me was Magda Kolanka, who is from David Sharp's lab at the Care Research and Tech.
Yeah.
CRT.
CRT.
Yeah, we've already defined it, so you can use the acronym now. It's fine.
Center at Imperial, who used who's kind of working on something very different to what we're doing, but using early detection systems, in this case a sleep mat, to be able to predict respiratory infections in people living with dementia, living at home, and to really enable with that, feeding this to the NHS and enabling people to stay in their homes for longer. And I thought that was just a really nice, it's such a different approach, but like using data for common good. And we had a lot of discussion about it afterwards in terms of data analysis and training, something that sounds very scary, AI systems, but like, how can that best be done, and what uses do they have, and how do we actually do science? So I thought her talk was a really good jumping off point to maybe have other discussions as well. So that was one of my kind of one of my highlights, sure. And if you do parallel sessions, you can showcase double the amount of talent in a way, mathematically. So I think we'll do one last whip around about highlights. So Beth, final comments.
More comments. So I was genuinely very impressed by all of the flash talks. I think sometimes you can go to a conference and they say flash talks, and they said it was 1 minute, and I thought, how can anyone describe what they've been doing in one? Because 1 minute is really short. I've been to them where it's 3 minutes, and I'm like, okay, 3 minutes, it's a good amount of time. But 1 minute, and I thought, how are they going to do this? And every single person explained their work in such a succinct manner It was clear, it was concise, they were engaging. There were a couple of little "ooh, come and see my poster if you want to actually know what we're doing." But I just think all of the researchers did such a good job of that. And I think it's the best flash talks I've ever seen at a conference. I was really, really impressed. And then it made me want to go to some of the posters. I saw somebody who was reaching Gaia Brezzo from the University of Edinburgh in the same institute as me, but you don't always know what people are doing. And I really loved the project that they were working on. So I then went and spoke to them about it, some of the models that they've been using. And I found that was a really good sort of starting point to chat with people about their work. So the flash talks were done fantastically.
Yeah, they were really good. Did you see the one done by Abs that was he took a bit of a different approach where he asked the questions to the crowd.
Yeah.
And I think yeah, different to, I think he just worked that format very well rather than sort of squeezing the PhD or whatever into 60 seconds. It was just perfect.
It stuck, didn't it? Yeah,
It
it was really good. It was really
Yeah.
good.
So for people who didn't see it, do you want to just briefly say what Abs did and maybe where he's from?
So Abs, this is slightly biased. He's also from CRNT, but he basically asked a question of how many people in the room think the EEG could cause accelerated aging, and people put their hands up, and then he asked about different factors, and gradually the hands went down, but some people still thought that they might affect it. And then he was like, well, you guys don't need to come to my poster, but everyone else come along. And it was just perfect, I thought. But also, a really impressive bit of work from him, and looking at how these EEG signals can predict accelerated aging. And yeah, just super 15-year long data set is it's just very, very compelling data, and a bit different.
Yeah, it did stick in the mind. It was such an interesting approach to it. It was brilliant. Yeah.
And very efficient. Part of it is very much very different than any other flash talk, but hit it on the nose, and all within 60 seconds. I was the timekeeper for that flash talk session, and I have to say, I was really worried they would run over boards. We were very strict with them. Not a single one. And a lot of them, I think, because I scared them a bit too much, stuck to 30 seconds. I was like, no, so you can talk a bit more.
Yeah. Yeah, you really embedded a lot of fear, I think. In them.
Oh God, we don't need to have this on the air. Okay, Dane, one of your highlights.
Okay, so I'll try and swiftly move it on. I apologize. That's funny. One of the other talks. So again, I thought the sort of parallel sessions were brilliant. But one of those from one of the parallel sessions was one of the first parallel sessions, the exchange auditorium. It was specifically Amanda Hesselgrave, who was talking about the biomarkers factory. Yeah. And she's just, her style and delivery is brilliant. And she's just seemed relaxed and knowledgeable at the same time, which was a brilliant mix for the platform that she's trying to push forward, the biomarker factory platform, which we have access to, which allows us to interrogate different systems with different biofluids, etc., that we can look for different markers in. But because she's so sort of knowledgeable and laid back, you feel like it's an approachable thing for you. She showcased the work, she showcased the abilities. She also highlighted some of the current caveats that were there, but delivered it with what we can talk about, how we would build this up and how this might work for you, for your particular samples, and how we might push forward with that, and the current strengths and maybe where the gaps in their particular data sets are currently. So I just think overall, whilst again showcasing the capabilities and, like I said, the caveats, I feel like a lot of people might have been inspired by what is possible, and also have felt the ability to be able to go, right, well, I can just go up to her and chat to her about what we can do moving forwards. And that can only be good for a platform that's supposed to be utilized by all of the different teams across the DRI. So I just think it's really good. And I'm a big fan of the sort of work that she and Henrik Zetterberg are doing at the biomarker factory. So I think the delivery of that was spot on. So it was really good.
Oh, that's good, because I think it's one of those weird things when you have platforms and researchers, and you somehow want the platforms to be very accessible, and yet. So I think it's a definitely tough sell she was trying to do there. I had another highlight from actually the same session. That's something that my team came back, and we just did a kind of highlight session in the lab meeting yesterday. And they all really enjoyed Zana's talk from UCL. So she's she's involved with the human tissue hub that the DRI is now setting up in combination with Colin, who's up in Edinburgh, and Susanna's down at UCL. And I think she gave a really interesting talk, coming kind of from the pathologist background of these are the different aggregates, and she showcased the digital pathology they're doing, and the most recent preprint of kind of scanning in a lot of archival brain tissue and analyzing that. And I thought that was something that inspired my ECRs and the team a lot, and we've been kind of having long and hard discussions about how we do neuropathology and how best to approach it. So that was great to kind of get inspired students.
Good. Inspirational. I love it.
Inspirational, yes, exactly. Which is, I guess, ultimately the entire point of Connectome. So before we close, let's zoom out a little. What do you think were the hot areas of discovery? What was the big takeaway? And in your own research areas, to maybe bring it down a bit more, what do you see as the next big challenge? Where are we moving towards? Anyone want to go? Yes, go for it.
I'll keep it brief, and then we can hear from there. I'll just give you time. I'll just, I think, like I said before, I and a lot of other people that I spoke to really liked the sort of breadth of going from basic science to, drug discovery even, or testing of drugs, to hearing from individuals with dementia. And so I think one of the big things that came over to me was the idea of translation and translational science, and even to a degree, although maybe to a degree it is applied science. So the idea of trying to get our work out from bench to bedside. So trying to see what we can do with the research that we're doing. And it just came over well as to not have any one stage of that have too much emphasis. So you can see how basic science can be translated to identifying the best targets, and the best way to then stratify those targets, and the best way to identify the best therapeutics for those targets, and then execute it. And perhaps we obviously need more of that execution part, but for me, the way that it was styled, it was a doable, visible thing. And I suppose the block to that at the moment, or one of the blocks to that at the moment in for instance, Parkinson's research is the idea of when you would test those things. So we currently have the ability to identify individuals to a fairly good positive degree once they have the actual manifestation of the disorder. But quite often, perhaps we're working on model systems that predate that are the precursors for that. And so translation of our drug is perhaps not working when we do take it to clinic, because it's at a different stage of the disorder. And so I think getting biomarkers and ways of identifying, again, let's take Parkinson's earlier, is going to only help us to translate, because perhaps we can work towards using different identified therapeutics and tools at different phases, really trying to stratify it into different parts of the disorder. But for me, translation is one of the things that really was strongly coming across. And that's the next thing, is to work out how we get much more of the work that we're all doing to clinic to actually help the individuals.
Good point. Beth?
Yeah, no, I second that completely. One of the things that I really took from the conference, and I think probably because the last session, sort of ended talking about a clinical trial, was, how do we get to a clinical trial, and how would that work? What's the best way to have the clinical trials, and how do we get the most out of them? And obviously, Sarah Tabrizi's talking about going from discovery and all of that, basic science work to the actual gene therapy clinical trial, and that sort of leads on to in my sort of area of research in cerebral amyloid angiopathy. There's actually the first clinical trial happening for CAA called the Capricorn trial, and it's for both individuals with Dutch type CAA, which is like a familial version, so genetic, and then also sporadic CAA. And it's like an RNA therapeutic to reduce the production of the protein that produces amyloid within the brain, and then which obviously goes to the vessels. So this is super exciting, because in this study that they'll be extracting CSF and looking at species of amyloid within there. And I think it's a really exciting time for the CAA field specifically. And I think it really hits on these hot areas of discovery, and how you can go from sort of basic research up to clinical trials.
Sure. Tom, what do you see as the next big challenge or opportunity?
Well, I guess for me, I think the emphasis around lived experience participants coming to the Connectome, I think it's the inclusion of people with lived experience in designing, particularly in my work where I'm trying to design a home testing device, and actually really considering the co-design process and how to make people's thoughts feel validated and listened to, and really help that drive the sort of, I guess, the design and development of my device, but then and see how that affects the progress being made in these projects, and then I guess how that then translates into commercializing these things so that they do get to bedside. That was my big sort of takeaway and the theme of the whole conference. I really felt that.
Yeah, no, I think I agree, and I think that was, I guess, ultimately the intent of the conference, and I think it was executed beautifully in that way. So yeah, even for the fundamental researchers, not basic fundamental researchers.
That's the better word. Thank you.
Oh, you're welcome.
I prefer that. I prefer it.
Just when you say like foundational, because I'm like, I do basic science.
Yeah.
It sounds just a bit naff.
I always think it hits the ear wrong as well. Yeah.
All right. Well, I think that wraps things up for today's episode. Huge thanks to everyone at the UKDRI for putting together such a brilliant event. I think we've been definitely highlighted some of the highlights. And of course, thank you to our great guests, Beth, Dayne, and Tom, for joining me today and sharing such insightful reflections. See you next time. But for now, I'm Anna Mallach, and thank you very much for listening.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk